Exactly. I was like, it's still. But it was at least like one time. That's nice. Yeah. We have an appointment for lunch with Foster now. Yeah. Because his daughter's like hers. It'll be covered by the program, so I can see. Well, they scheduled an appointment for me. Really like she's like sorry herself. But now I never used the laser pointer.
That won't show up on your Zoom. The vast majority of people are here. That's all.
How are you doing? So it was just like. Not teaching, but today is almost a book of ladders. I like this thing already. Exactly. So I like I'm kind of double booked. Like I should just
Yeah. Hi, everyone. Let's get started. It's my pleasure to introduce Tiffany Lowe-Power from the plant pathology department. So she joined in 2019 in the fall, which was a great time to join. And so she did her undergrad at Georgia Tech and then went to University of Wisconsin Madison and from there to Berkeley before coming here. And I think we're gonna hear about Ralstonia. So mostly, I guess your lab works on.
Bacteria-led pathogens. Yeah. And this is the latest thing we've been working on. Okay, so I think I start with some disclaimers on this talk. Oh, no, I don't. I start with some of why I'll be a little frazzled. We have a whole bunch of talks going on in our lab today after my seminar. And then also Stratton will be giving his exit seminar on Monday. So it's kind of like, I don't know, there's like an intermission over the weekend. But if you get really excited about Ralstonia bacteria and you want to see some of the undergrads in the labs talk about different molecular traits, or postdoc Natalie talk about transporters involved in Ralstonia, or Stratton talking about intermicrobial warfare, there's more than you probably really want to dive into. But we're excited. And the second part of my disclaimer is that what I'm talking about is pretty new to us. We kind of discovered some interesting biophysics in December, and we're still trying to get our heads around exactly what we're working on. So this seminar is a really good opportunity to try to build out the narrative, but it might not be logical yet. And so since it's also informal and you guys are like my neighbors, UC Davis folks, please feel free to interrupt and ask questions. I'm not sure that I would get through all these slides anyways, but they're useful for helping understand the story. And then I also want to hand out note cards in case anyone wants to write down any ideas or feedback. You can just like drop them off as you come up. If you're on Zoom, you can send me an email with some ideas. And yeah, so with that, let's talk about Ralstonia. And I've already stopped the video. So Ralstonia is a bacterial plant pathogen. These are bacteria that typically are infesting soil and then will gain entry to plants via wounds or natural openings. If you guys were here a couple weeks ago, Nuria such as Cole, gave a really nice seminar on some plant defenses against Ralstonia. Our lab, probably a little more than would be nice if it was a human pathogen. We're kind of cheerleaders for the bacteria. We're kind of interested in how they're causing disease, and we're really focused on that side. But you can see here with this infected tomato plant that this is a pathogen that causes a wilt disease. And that is because after it invades the roots, it gets into the xylem and blocks the plant's ability to transport that sap. So for today, I'm going to start with some work that is published, where we looked at how Ralstonia wilt disease is impacting the physiology, specifically xylem functionality in transporting the sap. And then the new part of our lab, which is trying to address this question of why Ralstonia are goopy. So I guess towards the end of my PhD, I was trying to think about, I think that this project came from writing the intro to my thesis. In terms of there's these papers and everyone talks about Ralstonia clogs the xylem vessels and that causes wilt. But when I'm reading the literature, I'm no one's ever actually tested this. And yes, there's a lot of bacteria in the xylem, but is that true? And I was reading some other, there's, beautiful microscopy in some diseases of these plant defenses called tyloses, where the plant's parenchyma living cells outside the xylem vessels will slowly grow these balloon-shaped appendages into xylem vessels and can wall it off. And then there's also some cool work about embolisms during disease, plant life. And I was kind of curious as to whether any amount of tylosis, plant autoimmunity, and gas embolism could be contributing to those wilt symptoms, as well as, of course, probably a little bit the dogma. There's a lot of bacteria in there. And some of why I was not sure that Ralstonia was definitely clogging the xylem is that, after 6 years of working with Ralstonia, I was deeply aware that it was a very drippy, goopy bacteria. This is a little bit sped up. I took this on my phone for fun. So this was not meant to be a scientific video, but it's only sped up a few X. They will drip. And so I was lucky to collaborate with Andrew McElron in viticulture and the USDA. And his lab has worked a lot with the advanced light source, a particle beamline up on the Lawrence Berkeley lab to use electrons to study plant anatomy. This beamline is set up to be able to take 3D X-rays of a sample. So it's many 2D X-rays as it rotates, and then you can make 3D reconstructions. And so Andrew was kind enough to share some time so I could put some Ralstonia infected plants on the beamline. And so I'm just going to talk a little bit about our results from that study. And really the credit for the experimental design here comes all to Andrew. I killed plants and brought some of the interpretation, but problem solving on the fly and getting this functional imaging assay to work was really from his expertise in studying xylem function. And so we were wanting to address the question, is the dogma true? Does Ralstonia clog vessels during disease? For this, we took tomato stems that were either healthy or had been infected with Ralstonia, and we excised a 2-centimeter chunk of them. We dehydrated the stems briefly under a heat lamp with the goal that during this dehydration, any of the xylem vessels that were sap conducting would empty out. And if any vessels could not conduct sap because they were clogged, they would remain filled with some substance. And then we imaged the tomato stems on the beam line. So this is one of the images that came out of this study. In this center photo is a 2D reconstruction across one of those stems. In blue, that little blue line is a transverse line that was drawn across this Z stack of the 3D images. And it's not great to see on a projector, but you might be able to see if you really squint and have very good vision. Part of what we see, so in the zoom in where I've adjusted the contrast, in X are xylem vessels that are filled with xylem sap. You can see a white ring. That's the lignin fortification of the xylem vessel wall. It's more electron dense. And then the more water cytoplasmic filled space tends to be lower density and is gray background. We do see some air-filled vessels in these dehydrated stems. Those appear as black because gas is very low density. And what was really interesting is that some of the xylem vessels that we infer are infected with bacteria had about the same electron density in their lumens as that lignin ring. So that was more dense than the xylem sap. And if you have the computer screen or you look at this photo, you know, at the right angle, when you cut down it, you can kind of see that. So this one here actually has like a bit of like some bacteria drip, and then you can see a more xylem sap filled vessel here. It's definitely a lot of like looking closely at the images and multiple, being able to go through the Z stack makes it more clear what's real versus an artifact. But generally we were able to see that Ralstonia does seem to be increasing the density of the lumens of some xylem vessels. And we can also look across many samples and count the number of air-filled vessels. So these would be vessels that mostly had been sap conducting and now have emptied out. And in the mock inoculated, these plants had, you know, approximately 60 to 80 vessels. The xylem vessels are on the outer of the stem, and they're arranged in these like not perfectly symmetrical vascular bundles, but you tend to have multiple parallel vessels together. And so we see that there's fewer sap conducting vessels during Ralstonia infection, and also like large swaths of that area where there's no sap conducting vessels. So those are probably areas that are really heavily clogged by the pathogen. And we have like some other microscopy that's consistent with this, and the microscopy is also consistent with the destructive sampling that many of our colleagues did before us to show, yeah, there's a lot of bacteria in the vessels. And so, you know, test the dogma. Dogmas exist for a reason. So it does seem to be that Ralstonia is clogging xylem vessels. But I want to introduce another dogma that's really common in xylem pathogenesis. I'm not sure if it's as common for the plant biologists, but we often read, and therefore we often say, that the xylem sap is very nutrient poor. And this is a great straw man argument. I used it in the framing of one of my papers in my PhD. Everyone seems to agree it's nutrient poor, and you know, compared to the phloem, there's less in there. But is that actually nutrient poor or more reasonably nutritious? And so in my PhD, I sent xylem sap that was from healthy plants and Ralstonia infected plants to the UC Davis Metabolomics Center. I wasn't here, but I was like, hey. And they did relative quantification of their primary metabolites. And we saw that there was some enrichment of some potential carbon and nitrogen sources. And moreover, when we grow the bacteria in the xylem sap from healthy plants versus plants that already had a lot of Ralstonia in it, even though that really infected plant was full of bacteria, there was more remaining nutrients in that xylem sap that the bacteria were able to eat in in a petri dish. And so, so we see a little bit, Ralstonia does increase some of the nutrient availability during infection. But my work was only relative. There was no absolute quantification. And some really great work by Caroline Baroque at INRA in France. has been like really building a really beautiful case against the notion that xylem sap is nutrient poor. So she used a different sort of chemical analysis, NMR, which are able to more easily get absolute quantifications of the nutrients. And she precisely measured amounts of 15 amino acids, 2 sugars, and 3 organic acids over time during bacterial wilt infection, got a sense of the kinetics as well as the total amounts of these things. And then in a second paper, she used that information to look at the metabolic reconstruction of Ralstonia to get a sense of how fast it needs to move different carbon and nitrogen sources through metabolism, and also did some further quantitative metabolomics in culture to really benchmark the flux rates as Ralstonia uses different nutrients. And then after that beautiful work, she made a mathematical argument that especially with the flow of xylem sap, able to continuously deliver this 9 millimolar total carbon, there is sufficient nutrients in xylem sap to explain the explosive amount of bacterial growth that happens. And so I especially want to highlight this because people keep citing Caroline's papers and saying, once again, xylem sap is nutrient poor. And so sometimes dogmas are just really hard to kill. So the xylem sap is not nutrient poor. Especially the flow of xylem sap helps bring nutrients to this bacteria. And yet we also see that Ralstonia turns off that flow. It clogs the xylem. So that's, a paradox. Why would Ralstonia make all this especially the exopolysaccharides? Like the cells make sense, right? That's bacterial fitness, division is good, but they're also making a whole bunch of this exopolysaccharide. And that's contributing to the xylem blockage and it's cutting off their food supply. So that's, they're shooting themselves in the foot. Why then are they goopy? So does anyone have any questions before I go into the next part? Cool. Especially since I think I probably will run out of time, so pull me off the stage. I want to start with the summary and then see how much explanation I can teach. So what we think is the answer to why Ralstonia are goopy is that this exopolysaccharide EPS1 confers some unique mechanical properties to Ralstonia's biofilms. And I see Hari in the audience. He's our collaborator who helped us measure these properties. We'll be talking about that. But this EPS seems to be mechanically unique. It has, Ralstonia biofilms are viscous dominated, which is in contrast to almost every other bacterial biofilm that's been published, which are elastic dominated. And so we think that this low viscosity and especially in viscoelasticity is allowing these colonies to spread via some sort of collective mobility. We're trying to come up with what terms we should be using for this. So if you have ideas, there's been some talk of motility, but maybe that needs an ATP motor. And so maybe mobility is better. That's a difficult thing. But whatever this trait is called, it allows Ralstonia to rapidly disseminate in tomato xylem. Once you put the bacteria in the xylem, they can clog the xylem and cause wilting symptoms within 3 days, which is faster than any other bacterial or fungal pathogen that's been documented. So they're really fast at moving through that system. And then if we look at the genes that are involved in the production of this exopolysaccharide, it is present in all of the plant wilt Ralstonia, which is this big green block. And then these are other Ralstonia that have different ecologies, and they only have very low homology to any of these enzymes. And so this trait seems to have come in somewhere ancestrally to wilt pathogenesis emerging, and possibly is causal for that pathogen phenotype.
When they cause the plant to wilt and die,
Yeah.
where do they go?
So throughout disease, they will continue shedding out into the soil. They're really good at surviving in soil. Most of their reproduction, is only gonna happen in the plant, but they kind of have that, pathogens can have different, selective behaviors. And I think of Ralstonia like Ebola. It's a very acute, fast, highly replicating pathogen in there. And that builds up a lot of inoculum that hopefully finds its way to another plant. A great question. So this is a slide that shows what this exopolysaccharide looks like on a petri dish. So we've got our wild type bacteria here that are making the exopolysaccharide. On this one, we have a mutant that's blocked. It has a deletion in one of those EPS biosynthesis genes, and the phenotype is that they don't end up making any. The pink is just a color indicator that we add for reasons I won't explain. But you can see this kind of drippy, goopy phenotype. This phenotype is really obvious. If you grow the bacteria, they're dripping. So it's been studied since the 1960s. And the genetics of this production were worked out very early once genetic engineering was possible, and gene sequencing, so the 1990s. They were able to make random transposon mutants that they mapped to this EPS biosynthesis cluster. And then when they infect plants with wild type or this EPS mutant, it's obvious that EPS is a major contribution to their pathogenicity. We have time over here, so this is days. And on the y-axis, we have the percent of the plant leaves that are wilted. And so you can see that disease progression. And the EPS mutants lower. Yeah.
I want to ask, is the EPS only secreted in planta, or is it also secreted in soil, and may that help it survive in soil?
I love that question. So it is, it's actually regulated via a really beautiful system where it's really only making it in the plant. It's dependent on quorum sensing, which bacteria use to sense their own cell density. And so they turn this on in plants. There might be some residual goop on them when they ooze out into the soil, and maybe that has some protective benefits to them. But it's always hard to I don't think anyone studied traits of root exit and stuff, because one, below ground is hard. And two, it's just there's a lot of stochasticity before you would get
Do
to
they
that.
get to the roots?
So they usually enter via roots, and then but then I think the actual network, that they go into the stem, and I think there's a lot of root decay and stuff if you work with them. So yeah, I'm not sure that they're making a conscious plan about where they're going, but they kind of get everywhere and try to eat the whole plant from what I can tell. Go on.
So while you have this picture up, I wonder about the distinct borders between Goopy. So that seems to suggest that goop avoids goop if it's other goop.
There is some amount of that, and I was actually just sending an email about this as I was looking at the slide again. So there is, but also no. So each of these red dots is going to be where a cluster of colonies originally were growing. The red is from tetrazoleum that we add to the media. And when bacteria are respiring, I'm not sure if they're adding electrons or removing electrons from this compound, but they change its redox state and it becomes from colorless to a red precipitant. And it's a marker of where the bacteria is, bacteria have been. So we tend to use it just to keep an eye on flaws, there's some mutants that will randomly look different. So that's usually why we have this tetrazoleum. But it's nice here because we can see that this one right here was probably at least 4 colonies that merged early on. And then, kind of, made some red precipitant, and like, there has been some merging, but then, yeah, there's clearly like these valleys. So Hari and then Steve Lindau both kind of brought up biosurfactants as being some sort of explanation, where there might be some biosurfactants that, biosurfactants can apparently cause this phenotype, and I learned about it an hour ago. But yeah, there might be something really cool there. So one more dogma to introduce about Ralstonia is, and this is common for like anything, like any trait. It seems like anytime biologists are studying a trait, they're like, it is metabolically costly, so why does this exist? And you know, and to some extent, right, everything does have some cost. And you know, Ralstonia is making a ton of this polysaccharide. And sugars, like that's clearly carbon. There's also some nitrogen here that is metabolically costly. And there's of course support for this dogma. There was when the Ralstonia metabolic model was first constructed by Remy Perrot, he modeled and then tested physiologically the energy cost of producing this EPS. And in liquid minimal media, there's a 22% growth rate impact where the production of EPS slows the bacteria down. So, you know, metabolically costly. So why make it? We don't think it's to clog the xylem because that doesn't make sense for nutrient availability. And like usually pathogens aren't wanting to cause symptoms. That's just what they do as they're trying to do something else. It's possible that EPS can help retain moisture. That's true of a lot of bacterial polysaccharides. They're hygroscopic, so they can both grab out of like the ambient humidity as well as hold on to the water and protect the cells. That's also the retaining moisture is why like soil health is all about cultivating a good microbiome, because that biofilm and polysaccharides that they produce end up conferring useful traits to the soil that the water retention. So it's not just sand where all the water is like dripping down into the wherever it goes. Polysaccharides are sticky, so it's feasible that this could be used for attachment. That's known for some bacteria. They're making this like visco gel that will slow down diffusion of some things, including nutrients, but also some toxic chemicals that plants might be deploying at them, phenolics, etc. Polysaccharides can also be just a physical barrier that ward off microbial predators. So bacteria tend to kill each other. Phages are viruses that infect bacterial cells. And then amoeba in soil can eat bacteria. And polysaccharides and other systems have been shown to, physically block some of this activity. But our model is that EPS1 is allowing Ralstonia to spread. And so the reason for this is chance encounters. So Hari and I met in like new faculty orientation in 2019. They had us introduce like a few words about ourselves before they got to how merits work. And Hari said that he studies what happens when goops flow. And I got excited and was like, I'm going to talk to that guy at the coffee break and get something useful out of this. And so we decided to team up. We wrote a couple small seed grants and eventually got one funded. And so And so the reason I was really excited when Hari said what he does is, I've been working with Ralstonia. This one is real time. So this is an inverted petri dish, and you can actually see the colonies. They will slowly coalesce, and when the physics is just right, you'll get that droplet of bacteria blubbing off. And so, I told Hari, I have goop. You have math. What can we figure out? And another thing with Ralstonia is a classic diagnostic of whether a tomato plant's wilting because of Ralstonia versus a fungal wilt, or I guess, desiccation, is to cut the tomato stem and submerge it in water And then after a minute or two, if it's Ralstonia or another bacteria that's abundant in xylem, but Ralstonia is one of the few in tomatoes, or only, you'll get these like threads, that like, and that's actual bacterial goop, that's kind of oozing out. So it's like, we have goop, this seems cool. Since I just mentioned the diagnostic, you will likely not find this in California. Ralstonia likes wet soils, so it's not really in California. If you have like tomato wilt disease, it's gonna be a fungus. So Hari taught me that bacterial biofilms are a classic viscoelastic material. And we can break down viscoelastic into the two components, one of which being viscous. And so what a viscous material will do is when force is applied, across it, it will stretch out and begin to flow. And then when that force is removed, it'll be stretched out still. And so if you're a bacteria, that can allow you to make a larger colony, but you might easily be removed, depending on what that force is. If you make a very elastic biofilm, then under that force, there'll still be that stretching, but there'll be the springing back. And the net effect, if it's perfectly elastic, would be recreating the original shape. So that elastic property will create sticky colonies that are resilient to physical perturbations. So the biofilms in your drain in the sink are probably very elastic, because every once in a while there's an intense force and they kind of want to, the organisms that are going to stick around are the ones that can. And so they'll be very elastic. And then, of course, biology is rarely absolute. It's usually got shades of gray. So most bacterial biofilms are viscoelastic, where they have some combination of that stretching behavior, springing back behavior, but also the viscosity will have allowed that footprint to have spread out, even though there is some stretching back. So you might get a larger colony that's also holding its ground. And so depending on the viscoelastic properties, in the same flowing environment, you might get a bacteria that creates larger biofilms because maybe that springing back ends up allowing it to build up higher. Or if you're really viscous dominated, you might get biofilm migration via that flowing process. So we wanted to know what the mechanical properties of Ralstonia biofilms were. We were just curious. We wanted to bring together some expertise. So on the biology side, we chose 3 Ralstonia that are representatives of the different plant pathogenic species. So we had some genetic diversity. We also decided to use Xanthomonas as a non-Ralstonia control. Fun fact is that it makes xanthan gum, which is a common thing in cooking because it's a viscoelastic material. So in Hari's world, xanthan gum is a model polymer. And so we're like, yeah, let's measure Xanthomonas that makes that in its colonies. There is some natural variation in the colony size amongst these groups. So we're like, I bet they have different numbers. They're probably slightly different viscous because some spread a little bit more and others are more dome shaped. More dome would be, some viscoelastic properties. And so PhD students in Hari and my lab worked together on this project. So Matt carried out the biology, grew our colony biofilms, scraped them into a test tube to take to Hari's lab, and together with Value, developed systems for measuring the biophysical properties using this cool instrument called a rheometer. So this has a stationary plate at the bottom. The red is our bacterial sample, and this is a rotational plate that's hooked up to some really sensitive sensors. It rotates at very specific forces and measures the drag forces back up to that sensor. And apparently you can measure all sorts of properties depending on how you rotate that sample. And from that, we calculated some viscous and elastic properties of these bacteria. So our hypothesis is that, like all other bacteria, these two bacteria make viscoelastic biofilms. So Hari's group is able to separately estimate the elasticity and the viscosity. And it's common to take the ratio of the two to get a sense of if you're more viscous dominated or elastic dominated. And in yellow here are the values for Xanthomonas. It was extremely elastic and extremely viscous. And if you take the ratio, it is elastic dominated. And Ralstonia, as we expected, was much less viscous and much less elastic. And we're like, cool. We're gonna write up a little small paper. We found some things. We measured some things really precisely. Awesome. And actually, so this is the December thing. We got together in the room and then realized that the ratio was actually different for Ralstonia. They are actually more viscous dominated than elastic dominated. And so at this point, we're like, that's pretty cool. That's the opposite of Xanthomonas. We have an A and a B. There's different biology. And this made sense from Matt's work to scrape these colonies. So like Ralstonia will drip down into the tube, like left. The Xanthomonas, he kind of has to keep trying to push that in with the glass rod, and it's like sticking up there. And so, you know, it's like, what's known about other bacteria? There has been a lot of nice reviews and nice mechanistic work on this in other bacteria. And so we compiled the information. On the bacterial side, there is a bias towards measuring clinical biomedical bacteria, but also some model and some soil bacteria have been measured. They're pretty taxonomically diverse. People have done this approach for a few yeasts, probably because they also look a little goopy, and so that's interesting to potentially measure. And a little bit with some natural biofilms isolated out of streams. And universally, in all of those published reports, all of those biofilms were elastic dominated. And so Ralstonia was different. So one of the things we did was wonder if we were wrong. Like, thank goodness we measured Xanthomonas, because we're like, okay, we're not just doing all the math wrong, or all the measurements wrong. We are able to measure an elastic dominated thing. It's not just us. It might be biology. And so, you know, we think what's going on is that this is, you know, it's consistent with the picture, it drips. And maybe this is a useful adaptation that these bacteria have. By being viscous dominated, they might have more flowing and migratory behavior under force. And so I'm a bacterial geneticist. I like testing mutants. And luckily, a lot of folks have already created some of these mutants. And some is known about what's in biofilms. There's the exopolysaccharide. As cells lyse a little bit as they grow, extracellular DNA also becomes a component of this matrix. And there's some secreted proteins, including some like lectins that would, you know, non-covalently cross-link polysaccharides. I'm not going to talk about the extracellular DNA or the lectin proteins because it may slightly change the numbers, but it's not what's conferring this unique viscous dominated behavior. It's the exopolysaccharide that's responsible. So when we look at the ratio for a mutant that does not make exopolysaccharide, instead of being viscous dominated, it has a ratio of about 1. And this is consistent with how the samples look in the tubes. Matt described it as having the consistency of acrylic paint. It was, you know, difficult to get this amount. And so I didn't want to bring all the physics graphs, but the overall values of the viscosity and elasticity is that when this EPS is being produced, the viscosity and elasticity have been massively dropped. The EPS mutant, as you can see by how sticky it is really viscous and elastic. So that'd be like the base state of a cell that doesn't make this polysaccharide. There is a little bit known about this polysaccharide. In the 80s and 90s, like a large amount of analytical chemistry was thrown at this molecule in this paper, and they solved the structure here. It is a trimer of three sugars that are kind of weird and rare. One of those is common enough that people have decided to call it, GalNAc, N-acetyl-galactosamine. But these other ones are really weird, and especially this one is just a weird one that is not common, at least in the literature. And if we look at the structure, over here I've drawn put out cellulose, so it's a polyglucose polymer. Cellulose has 3 hydroxyls as side groups down each sugar chain. There's relatively few hydroxyls on Ralstonia's polysaccharide, but there's a large amount of N-acetyl groups, as well as just a completely reduced carbon on that top one. So there's not even an oxygen there, so that's like pretty nonpolar. And there was a seminar on biofilms in plant pathology on Monday, and he was studying biofilm, and he mentioned that acetylations on polysaccharides are known to influence the stickiness of polysaccharides. So, I mean, I don't know, hydrogen bonding, you can kind of look at that, I don't know. I don't think more about that, but. So one of the next things we asked was just a question we really like asking in the lab these days, is how conserved are these genes across plant pathogenic Ralstonia? So we used a comparative evolutionary framework. On the bottom of this species tree are the 3 species of the wilt pathogens. These tend to be infecting plants. That's probably the only place they really grow. But then they can also survive in soil and surface water to transmit between hosts. At the top there, relatively fewer genomes because they're not causing as many problems in the world. They have much more diverse ecosystems. There are some that are plant commensals, some that live in soil, surface water. A lot of them also are great in potable water systems, and including can form biofilms on the filters. Because of that, they end up in 16S microbiome studies as a kit contaminant. They also can end up in IV solutions that is, if it goes to an immunocompromised person, can lead to bacteremia. And then 2 lineages have become permanent residents of the water recycling station on the International Space Station. Like the clonal branches of that top thing are like isolates that were sequenced each year. And so we're curious about, what's the presence of these exopolysaccharide genes in these different relatives. This is like common for bacterial exopolysaccharides to be produced by a genetic unit. There's transporter genes here as well as enzymes that make each of those components and some regulators. Tabitha in our lab did blast searches across these genomes and visualized it across the species tree. And we see that there is high conservation of these genes in Ralstonia wilt pathogens. If you're eagle-eyed, you'll notice this one genome that doesn't have it. It might be a garbage genome, but we can't throw it away. NCBI says it's too small. But it's in a lineage that might be genome reduced, so like that could be biology, or most likely an artifact. And then the other Ralstonia only had sporadic low homology hits of any of these genes. So this trait seems to have, emerged at the, beginning of wilt pathogenesis as an ecological trait for Ralstonia. So what we know from the literature and the really beautiful work that was carried out in the 90s is that production of this polysaccharide is a social multicellular behavior of Ralstonia. And so that's where we get into the quorum sensing regulation. So that quorum sensing is common for bacteria, especially bacteria that live on hosts, to have this system to make special molecules that they then sense. If they only will sense them if the concentration is high enough. If the concentration is high, that means that a lot of kin are around to have made it. So they're able to regulate some group behaviors based on this trait. And it's been known that quorum sensing turns on EPS production from nothing to like gangbusters and is really responsible for this. And ecologically, Ralstonia is probably only in that high density state when it's growing on plants. And so we think that this is EPS1 emerged in the plant wilt Ralstonia and is conferring some collective mobility to them. Yeah, 5 minutes, great. So we tested this on agar. So we decided to adapt a cool assay in the literature where we grew Petri dishes flat, like you normally do, or at 39-degree angles. 39 degrees because we had an object in the lab that turns out to be 39 degrees. And so after 2 days of growth, the wild type bacteria have dripped down. The EPS mutant hasn't really moved. And so, you know, under the force of gravity, EPS1 is enabling this migration. We were curious about testing this dogma. So production of this EPS is costly, and in minimal media, it's been shown that that cost has an effect on the amount of cells that can be produced per amount of carbon and nitrogen. But the plant is not a test tube. Shaking culture will be moving all those nutrients around. And in a plant, there might be more diffusion. There's definitely more surface growth. And so, you know, is EPS potentially helping in a slightly more plant-like environment? So instead of doing this in plants, we decided to do on agar. We grew individual cells or grew individual colonies and then cut them out and just dilution plated the colony. Each colony originally starts from one cell and then grows up into a population. And the wild type bacteria actually have a higher, they're larger because they're also making that EPS. But they also have more cells per colony. So it's actually increasing their growth, potentially improving the terrain in which they can start diffusing chemicals. Maybe nutrients are diffusing through the polysaccharide better than agar. Maybe Hari can tell us. We measured colony area. I just want to get to this experiment that Natalie did in our lab. So we also showed that this probably is true in planta as well. So Natalie, instead of doing soil drench, inoculated individual strains into the stem. And then 3 days later, harvested the population at the site of inoculation, below or above. The wild type strain had really high populations in the site of inoculation and had already spread. And the EPS mutants was much lower. So that's cool. Seems to be true in plants. And I think that's, we're trying to also work with a former, he was like my PhD buddy, Tuan, now has a lab at University of Southern Alabama. He's got these cool microfluidic devices that he coats with cellulose that can better allow you to look at bacterial biofilms. Reaching out to some folks who have other Ralstonia in their collections to see if they also drip. We think no, based on what they said in the emails, but they're just gonna do the simple experiment. They don't have to do 39 degrees, they can choose their lab object. So we think this EPS1 we know it has unique mechanical properties that confer the ability to spread upon force This seems to be helpful for the pathogen's rapid dissemination in xylem, because of course that's a flowing environment with fluidic force. And this trait is ancestral to the wilt pathogenesis and absent from these neighbors that have other ecological roles. This work is a co-first authored work by Jiayu Li in Hari's lab and Matt Cuparweo in my lab. I don't even know if that's all the collaborators who just trying to, it's been like changing since December. So it's like, whoa, we found something cool. So with that, do you guys have any questions? There's also other people in the lab. This is old. I meant to update that. This is us from 2020. So.
Question slash future direction for testing mobility, like mobility idea. I wonder if you can like rescue the mutant by like using the EPS inside the plant and then putting mutant and see if they actually spread.
Yeah, I definitely want to try that. Putting it directly into the plant, like genetically engineering a plant to not be able to transport water, probably isn't going to work out beyond the fact that I don't know how to genetically engineer plants. Directly putting stuff into xylem is kind of hard. It's under negative pressure. And so potentially just by stabbing something in, you might be changing the physics. But I want to try to get funding to do that idea. And the thought is to take our EPS null mutant and try to get it to make other bacterial polysaccharides that have other physical properties and see if the the particular physics of this one, influences how they spread. But yeah, so their ideas. I'm gonna write a grant. So.
Supposedly the flow in the xylem is laminar flow, so that it's not very kind of fast.
Yeah.
Does this work if the flow is gentle?
So gravity's gentle. And it works with gravity. I wish Hari didn't have to teach right after this because like Hari knows about laminar flow and the implications of that. It actually, if anything, laminar flow would work better also for elastic polysaccharides. So from what Hari's told me, under the constant flow, the hydrogen bonds of the polysaccharides, they'll be like potentially more relaxing. And what's been shown in some more sticky bacteria is they make these things called bacterial streamers. You get this long thing, it still holds, but it's kind of this like spew, it's the drain ones. But if you have the sharp forces, that's where, so that's like a non-Newtonian fluid, like the cornstarch that you punch. All of ours do actually, these are all shear thinning based on some graphs I didn't show. So that means that under more force, they thin out more. And under less force, they thin out less, and there is some sort of plateau.
Wait, does the bacteria have its own motility? Do they move around?
Yes, it has two in addition to this. It has flagella, but it only uses flagella to get into roots. There's almost, to my knowledge, no bacterial pathogen plants that use their flagella in plants. And that is probably because so many different plants have flagellin immune receptors. Thus, they kind of really guard against that. And so Ralstonia uses it to find roots, but it also has this grappling hook pilus motility, where this pilus comes out the cell and then retracts in, and so it like drags them forward. That's known to help some with like movement in xylem. Twitching motility will only go against flow because it's at the pole, and so flow will make a rod-shaped bacteria like pivot, and so it can only go downstream. So maybe like twitching might go against the flow a little bit as like the blub goes with the flow. Cool.
Okay.
So I'm gonna have pizza with some of you guys. Yeah.
Yeah.
Is that so random?
Yeah.